Hi, everyone. Welcome to our webinar, Studying Microbial Communities, a review of EcoPlate use cases and the newest community analysis features in the Odin software. My name is Kasia Proctor, and I will be your moderator today. First, a few housekeeping items. So this webinar is being recorded, and we will share the link to the recording once it's available on our website. If you have questions, please submit them at the bottom in the Q&A chat window, and we will answer as many as we can at the end. Many of you submitted questions during the registration process. We will either do our best to answer those during the webinar or review them at the end. And now let me introduce our speakers. Dr. Enrico Tatti is our distributor sales manager. He received his PhD in soil microbiology from the University of Florence. And Dr. Max Gravener is our field scientist, and he got his PhD from the University of Georgia in microbiology. Welcome to you both. And Enrico, please take it away.
Thanks, Kasia. Can you hear me loudly and clear?
Yes, we hear you well, thanks.
And hopefully you can see me as well.
Yeah, go ahead.
So thanks for the introduction, and I want to say good afternoon from Florence, Italy. I know some of you may be in a different time zone, so I will like to add good morning and good evening, just in case you're following a different time. So it's a pleasure for me to be here today and present this webinar together with Max. We're gonna talk about one of our most important products in biolog, the EcoPlate, as Kasia already mentioned. And particularly, we will mention two main things: how you can use EcoPlate and how they work, and some recent example of their usage in scientific publications, and then how you can use our new data analysis software specific for EcoPlate in our instrument, Odin instrument. So let's start with an analogy. Microbial communities are the unsung heroes of our ecosystem. As they drive processes that sustain life on Earth, they're really, really important for these reasons. However, we know that understanding their functional roles has always been a complex challenge for microbial ecologists and anyone concerned. For example, in the recent years, we got a lot of new information on different environments, key environment, for example, the gut microbiota, where we know, for example, that there could be up to 500 different microbial species, or about another key environment like soil, where we know that one single gram can have up to 10,000 different species. And this is surely thank you to the most recent advances in DNA-based methods. We can surely name them. We can see, to which phylogenetic groups they belong, and we could follow how they change over time. But we still struggle to know actually what they do in their environment, and what is their role in the ecosystem where they live. So, if to make an analogy, it's like being in a room with many people that we recognize, that we know, but that we can't hear what they actually say. So imagine being able to decode the hidden languages of microbes in soil, water, or even within our own body, the gut microbiome. So we need complex and important solution that can help us understand what microbes can do in ecosystem and what they have to say about their functional and ecological role. And this knowledge, of course, is not just academic. It has a profound implication for agriculture, environment, conservation, and human health. So with this presentation, we will dive into how biolog ecoplates are revolutionizing our understanding of these microscopic powerhouses, the microbes, and why this analysis is crucial for us for a sustainable future. So let me start introducing how ecoplates works, but in generally the biolog solution, as I said, they have the same rationale behind. So first of all, biolog solution, they work on, they focus on a cellular approach that works on live cells. So this is, of course, very different from DNA-based methods where you kill the cells and extract the DNA. Here we work the cells that are alive. And the core unit of the biolog technology is something like this, a 96 well plates that you may be already familiar with. The only difference is that has a smaller well, ELF volume. But it's important to consider that in biolog solutions, each of these wells contain a different chemical, or if you want, a different carbon source, nitrogen source, sulfur source that could prime and probe a specific metabolic pathway. So the plates, they come pre-made, so you can, when they get there, you don't have to add anything. The only thing that you have to do in a very simple protocol is to extract your cells from the community you want to study, add them, and add an indicator for metabolic activity that is actually a redox dye. A redox dye that will form a color if there is actually metabolic activity in that specific well. So just to follow the animation, you incubate over time, and after 24 hours, 48 hours, or whatever time is needed for the community under study to develop a partner, you will then end up having what we call a functional readouts. Since each well contains a different test measuring a different metabolic pathway, you will end up having very important information on what the microbial community can do in terms of functionality. And actually, these slides is a little bit misleading because you won't have all the same color at the end of the incubation in all the wells. You may have different shade of color that will indicate the importance of that particular metabolic pathway for the microbial community under analysis, or the efficiency, so to speak. So just to give a little bit more details on how it's working, in each well, the microbe will find food and they will activate their catabolism to actually generate energy and then growth. And this is what the process that our redox dye actually captures. So it's a very simple chemistry. Our dye initially is a tetrazolium-based dye, is initially oxidized in a colorless form. And because of the microbes, they are metabolically active. If they are metabolically active in that particular well, they will eventually generate NADPH or NADH. And this molecule will reduce the oxidized redox dye. And in this process, the redox dye will form the color that you've seen in the previous slides. So very simple chemistry, but very important. Very important for the type of questions that can be answered. We can, for example, very simply, just looking at the end results, answer what are the functionality of a given community you want to study, or how much these functions are important, what is the intensity of this function, quantify the dye color reduction, which will be proportional to the actual metabolic activity, as I said. But also another important information that we can fetch from these plates, if we measure over time the color formation, is how fast or how efficiently these metabolic activity are present and ecologically relevant for the microbial communities that are under analysis. So we can answer what, how much, and how fast, depending on the level of resolution that we want, leveraging a very simple chemistry based on the redox dye and the different compound in each well. So as I mentioned, and as Kasia mentioned, this is with this webinar today, we will focus mainly on EcoPlates. We have a different solution that can be used to probe microbial communities, but EcoPlates is the most used one by our customers. There's plenty of publication, and I'm sure many of you that are watching now are familiar with this particular design. In EcoPlates, there are 31 different carbon sources. They belong to different functional classes that we'll see in the next slides, that are repeated 3 times. So actually there's a triplicate sets of carbon source. So the user can decide the design of their experiment, adding 3 different microbial community or the same microbial community after 3 different times, or or just use them as a technical or biological replicate. That's up completely to the user. And the peculiarity of these plates compared to other biological plates is that there are substrates that are a little bit more complex. And they have been divided here in these slides for you in different functional categories that are very important, as you will see in the next few examples that I will make. For example, there are compounds like carboxylic acids, carbohydrates, amines, phenols, polymers, and amino acids. And just to name a few among the carbohydrates, just to remind you, there are some compounds that are more complex sugars, not really simple, like celluloose or xylose, if you want to know those sugars that can originate from plant material. So basically, using the Biolog EcoPlate solution is as an excellent way to gain an holistic metabolic view of the communities that you have under analysis. And it's very complementary and very data that you can get to merge that with the data coming out from sequencing, like DNA-based technology. So just to make an example, you may want to compare two different communities, community 1 and community 2. And this can be the same community after time in a longitudinal study or after a treatment. It doesn't matter, just want to make a comparison. You can think about the two communities that they may be formed by different members, each, different microbial members indicated by the different letters. So community one may have A, B, and C, and D, and E, and then the other community may have J, K, L, M, P, Q. And so this community may be very different, and this is really important to unravel this using DNA-based methods, but they may also have the same metabolic capability. And this is the information that actually the EcoPlate can help to unravel. So if you look at the left here of the slides, there are the different functional groups. You can see the different groups, they have different functionality. And even if there are different microbial groups, they may happen that there's a functional redundancy because different groups may be able to do the same thing. So actually using EcoPlate will complement very well with data coming from sequencing. And also in case that you have metabolically equivalent community, you can also, as I mentioned, use the relative rates of color formation to distinguish them more. So again, it will depend on the different level of resolution that you will apply for analysis, and Max will talk about this later in this webinar. It will depend how in depth you want to go in the analysis and make it simple or more complex depending how you read. But again, the EcoPlate will focus on what the microbes can do and help to understand if different communities have the same capability, which is obviously important for ecosystem service, as we know. Okay, so I will now, before passing the ball to Max, I will talk about 3 different case studies that are very recent, have been very recently published in the literature, and they represent an excellent example on how EcoPlate can be analyzed and how flexible the information from EcoPlate can be in terms of the analysis you can decide to do. So in the first case study number 1, we'll talk about specifically on sustainable agriculture and the use of EcoPlates and other biological solutions in this important sector. So the paper is a recently published, the paper that I want to present is a recently published paper from Maciek and other co-authors based in Poland and published on applied soil ecology last year, where actually, just to make the story short, they had a soil, an agro-ecosystem, so to speak, and they added phosphorus that, as we know, is a macro element for plant growth and crop growth. And then they also added some biofertilizing strains on the genus Bacillus and Penibacillus. So after they made this addition, they studied the microbial community response using 3 different approaches: studying an enzymatic response to of this soil community, the composition using the TRFLP and NGS sequencing, and the functionality using biolog plates, and also using channeling the 16S data to the PICRUST database to gain a little bit more insight on the putative functionality. But what makes this paper very interesting are actually 2 factors. So what the user did, they didn't just use EcoPlate, but they also use our FF plates that are generally used for the analysis of fungal microbial community and fungal isolates. And they run the same sample in EcoPlate and in FF plate. With EcoPlate, with the goal of focusing more on bacteria, and with FF plate focusing more on the filamentous fungi. And the additional thing that they did in this very interesting publication was to read the plates not only at the wavelength that is related to the metabolic activity, which is the 590 nanometer, that these plates can be read to measure metabolic activity and the dye reduction, but also read the plates at 750 nanometer. And then calculate a ratio, which is a very important ratio to measure cell stress in the environment. This has been applied successfully to microbial community, but also on isolates. So when the results are showing a community that has a high metabolic activity but a low biomass value over time, that means that the cell in the community, they cannot convert efficiently energy into biomass. So the community is thought to be stressed. So when this ratio is higher than 1, there's stress at the metabolic stress on the community. While on the other side, if this ratio is close to 1, meaning the metabolic activity is then efficiently converted to biomass, then the community is not in a stressed state. And they converted this in heat maps where you see warmer color for generally speaking, you have a higher value of this ratio. And when you see colder color, it means that the value is closer to 1 or even lower. So this is a very important, and they look this for both bacteria and fungal community. And this is a more close up of the results. And you can see on the top the results coming for bacteria communities, and on the bottom the one related to filamentous fungi. Very interestingly, with this analysis, it was possible to see that the most stressed community over time, you can see going from left to right, the time changes, a few months after the addition, from few weeks to a few months after the addition. The microbial community, the bacterial community is initially quite stressed on some specific function. So this is also what Biolog can help you in telling you what functionality are stressed, not only that the community is stressed. And then this stress seems to fade out over time. While there's not a clear pattern in the fungal community, there's a certain level of stress here and there, but there's not a clear pattern like the one for bacteria. So obviously the bacterial community was more perturbed as expected, adding biofertilizer and phosphorus than the filamentous fungi. And these data, looking at also NGS was showing a change of both the fungal and the bacterial community at the same time. But then running these data sets against each other for enzymatic data analysis and PICRUST for the functionality of 16S, the 2 data sets were agreeing with the data in Biolog. So very important paper that's showing the sequencing and Biolog methods and also enzymatic method offer complementary data sets, and they are both needed in this type of study where sustainable agriculture is the main focus. The second case study that I would like to present today is an example instead of how EcoPlate can be used in anaerobic communities. And this particular example here is related to sediments communities. And this is a paper from a Chinese group of Qinding published in March 2020, so in the middle of the pandemic. And really interesting of this paper, there are two factors I want to highlight. So first of all, we are studying anaerobic community. They are really important for ecosystem process, the one in the sediments. One of the sediments are communities that are known to have an important role in dealing with the excess of nutrient coming from land. And so they have a filtering, functionality to avoid a lot of nutrients. They go in the ocean and form this algal bloom. But the goal of the paper was to test the functionality of the community, the anaerobic community. So the plates were used in a microclimate study, but in anaerobic condition, and the EcoPlates, they work in anaerobic condition, to see how the community would respond to different time of exposure to this cyanotoxin that is produced by algal bloom. And what the user found out was really interesting. Again, I want to highlight that we are looking at an anaerobic community. They use an interesting metric that Max will explain more in details, the average well color development. Some of you may be already familiar with it. It's used often with the microbial community. That gives general ideas on the activity of the community, taking in consideration all the different functionality. And you can see that increasing the time of exposure to the cyanotoxin, there is a proportional decrease to the general functionality of the community. But also what biolog can do, and this is something that, the change in the community was also seen by community analysis 16S, but also what you can get, the higher level of granularity you can get is that you can break this down in this average well color development can be broken down in the different functional guilds or functional categories of the compounds that are in the EcoPlate. So you can actually see what are the functions that are most affected by treatment, like the presence of the cyanotoxin. So again, a very high level of granularity that you can get with the EcoPlate if you want, and Max will talk more about it in his part. And the last case study I want to do is a very nice paper where EcoPlates were used to understand the mode of action of microbial biocontrol strains. So we are back to sustainable ag if you want, but a very nice paper that shows the flexibility of the community. So this has been published just a couple of months ago, so at the end of March, in Journal Applied Microbiology by the group of the Di Lin Luo, so again in China. And the authors were focusing on basically peanuts, and which obviously is a very important crop for the capability of nutrients that can give to our diet. But what they were doing, actually, they were studying the endophytes at the seeds level. And they wanted to see how endophytes that are known to protect the seeds from pathogen would influence the pathogen like Aspergillus flavus and Fusarium oxysporum. And together to this, they created a synthetic community formed by these endophytes, seed endophytes, and known biofertilizing strains, and they added them to EcoPlates where they were Aspergillus flavus and Fusarium oxysporum. Again, the metric used in this analysis is average well color development that will be presented more in details in a few minutes. And you can see here that the strain of Fusarium and Aspergillus by themselves on the EcoPlates over time, they reach high activity. But when the synthetic community is added, there's a loss of activity compared to the strains alone. And again, breaking this down not only functionally wise, but also over time, it was possible to see those substrate that both the biofertilizing strains and the synthetic community can compete with the pathogens. For example, it has been seen that the community that was added to the seed, taken from the seeds, and the pathogen overlaps and compete for asparagine, acetylglucosamine, and methylglucoside. And also that pathogen were weakened by this synthetic community to use glucose twin 40 and twin 80, so complex polymer. So again, a very high resolution analysis that can be done with the biolog EcoPlates. And this particular paper using not a complex community, but two different pathogens and see how they can be combated by fertilizing strain and biocontrol strain. So this is all for my part, and I will now pass this presentation to Max, that will drive you through the data analysis and experiment example on EcoPlate. So please, Max, take them away. The floor is yours.
Thank you, Enrico. So I'm going to start talking about the new community analysis features available on Odin that can help you do some of the analyses that Enrico had mentioned, as well as some additional features that can help you get more power out of your EcoPlates and the data that you can generate with them. So first off, I want to address the Odin family of instruments, just to establish a base for where we're running the EcoPlates. So we have two different instruments in this family so far, with Odin L and Odin 8. And the difference between these is simply the number of plates that they can run at the same time. And so Odin L and Odin 8 are the all-in-one solution for phenotypic characterization, microbial ID, and community analysis. These instruments allow you to simultaneously incubate and collect data from your plates, and will automatically feed that data into the software that will allow you to analyze it, which is the same software platform that you use to run the instrument. So it's all in one easy to use platform. And with Odin L, you're able to run up to 50 plates at a time, and Odin 8, you're able to run 8 plates. And the work that you'll see here was done on a combination of both. So EcoPlates on Odin, once you run the plates, you then move into data analysis. And the first step of this is curation. And so what curation allows you to do is to kind of do quality control on your data. And so a couple of things that you can do here are assess your negative controls and recognize any outliers that you might have. So you have a couple of different examples on the screen at the moment. Up here on the right is an example of a single plate on the curation view, and then this is 3 plates that were replicates of the same sample. And so we can see that the negative control here looks good in all 3 wells. So we have negative controls in A1, A5, and A9. So this could give you an opportunity to recognize that maybe you had some carbon source added to one of those negative controls, which may be confounding your results. And then when we look at the multiple replicate example here, we can see that one of the replicates sticks out in a couple of places. And so this black replicate here has a high signal in this well that's not consistent with the replicates of that well within this one plate. And so we may recognize that as an outlier that needs to be removed. And what you can do is pick and choose which of your replicates you want to preserve into the following steps of analysis. And you can do that just by simply unchecking the checkbox here. So the next step in the pipeline is the analysis tab. And so this is going to allow you to do some baseline analysis of your data. This is the same analysis that you could do with a phenotype microarray plate. Anything on our phenotyping license you can do here. And so this allows you to adjust your axes, export your raw data, as well as calculate a variety of parameters and do some pairwise comparisons. So here we have just the one sample again on the EcoPlate, and the values reported here are going to be the maximum height of that curve. And then up here we have a pairwise comparison example. So in pink is our reference sample, and then in blue is our test sample. And you can see that we can set a cutoff. In this example, the cutoff was 1 for the maximum curve height, and anything that meets that cutoff will be highlighted. And so this allows you to get a nice bird's eye view of your data. However, what's really important about the EcoPlates is the fact they have internal replicates. And so we want to be able to utilize those internal replicates for statistical power. And so then we're going to get into our new community analysis features. So in the community tab, the first thing that you'll see happen is that all the internal replicates for the plates will be averaged and displayed on in the software. And so this is the same data as the previous slide, but now we're looking at the composite of each of the internal replicates within the plate. And and we're still doing the test versus reference comparison here with calling out significant phenotypes. So there are a few different parameters that you can calculate here to be displayed on this chart right now. We have maximum height displayed, and then you can also do a couple different ones, including area under the curve, which is another popular one, as well as rate calculations or an average curve height, depending on what you're interested in. You're also able to toggle which wavelength you're using. Right now I'm displaying the 590 wavelength, which is primarily dye absorbance, but you can switch it to 740 as well, which will give you approximately biomass. So the next feature, which is the really new thing that we're adding to our analytical pipeline, is average well color development. So Enrico talked about this a few times during his case studies, but I wanted to go into a little bit more depth here. So what calculating the average well color development does is one of the things, it allows you to compare across different communities, regardless of whether or not you know the beginning cell titer of those communities or the component members of those communities. Average well color development gives us a holistic view of the metabolic output of your given community, and we can use this as a point of comparison. And the way that this is calculated, there are a few different steps involved. So first, we subtract the 750 signal from the 590 signal, which normalizes for background absorbance from, say, silt or biomass or other things that may have been in the suspension. And then we subtract the blank well, or sorry, subtracting the 750 from 590 normalizes the sample, and then subtracting the blank to get rid of that background signal. And then we average all of the color development across all the plate, across the whole plate, and that gives you the average well color development for that sample on that plate. And so we can see here is in this example of SN1 that color development really started to pick up around 17 hours and reached its peak at the end of the assay and had not yet plateaued. The next parameter that you can calculate is the what's known as Shannon's diversity index, or H prime. And what this allows you to do is look at the diversity of substrates that are utilized by your community. And this is really valuable in looking at the flexibility of your community to metabolize different substrates, and maybe whether or not it's primarily able to use only a couple of substrates early on, and then there are others, there are composite members of the community that pick up growth later on and allow it to utilize more carbon sources, or if it's fairly even from the start. And so this is calculated using a ratio of the color development in each well relative to the color development across all of the whole plate, and related as shown here to calculate this index. And lastly, the third feature is calculating Shannon's evenness index. And so evenness is related to the a parameter called richness, which is the number of substrates that have a positive response in the plate. And so you can adjust the cutoff or pattern development cutoff, what we call it, to filter how significant of a signal is required to call a well positive. And then that can adjust how the evenness is calculated. And so what this allows you to do is just have a general view of how effectively your community is able to use all the different substrates it's currently growing on, and if it's equally able to use those substrates or not. And so this is very typical for us to see a spike in the evenness index, indicating a uneven utilization of the different substrates, which will then fall off to around 1 as all of the different wells that are being utilized reach saturation. So now I want to get into a case study where I ran some different samples of soil from around the Bay Area on EcoPlates and used some of these features to analyze the data to draw some conclusions here. So the way that this worked was I collected some soil samples generously donated by Biolog employees from around the Bay Area. We see the locations approximate of each of those samples by these purple stars. And I took 5 grams of soil and mixed with some sterile water to collect the microbes, centrifuged them to get rid of all the heavy particulates, and then inoculated 100 microliters of that suspension into each well of the EcoPlates, and then incubated at 30 degrees for 48 hours, collecting data every 20 minutes. And so this is a pretty typical sample preparation that I've seen a lot of different customers use and works very well with these plates as they're pretty robust against different sample types. So getting into the data here from this geographical soil comparison, each sample showed very unique metabolic characteristics. For example, Palo Alto, which is over here on the right, was the least flexible community with the highest number of low signal substrates. So 10 different substrates on the EcoPlate gave little to no signal from the Palo Alto sample. And then the Cupertino sample was the most metabolically active. It was the first one able to degrade any of the substrates in the EcoPlate. After only about 12 hours, had the highest substrate diversity of any of the other samples. And pretty interestingly, none of the communities that I sampled were able to metabolize a couple of different substrates, including this phenolic acid, this 2-hydroxybenzoic acid, and the other one is the alpha-ketobutyric acid right above it, this carboxylic acid, which is pretty interesting. There weren't any real geographical conclusions that we could draw here, but it's always neat to see that there are different things going on in the microbial communities across different areas. So the next thing that I did with this feature was to look at native soil versus garden compost and to see if there were any impacts that we could visualize based on the addition of compost to different soils and the microbial activity there. So the first comparison that we were looking at was leaf compost, just a pile of leaf compost not added to any soil, then versus native soil. And I don't think this is probably surprising to anyone. The leaf compost was much more metabolically active than the native California soil. And so we can see that in a couple different parameters. So first off, we're looking at our averaged replicates here. So our negative control was negative for both of the samples. And then for each of the substrates, the blue here is the compost. The compost was able to pick up metabolism much more quickly than the native soil, and you ended at either a higher level than the native soil, or they ended at the same level because there was a lag for the native soil. So this is consistent with the presence of active biodegrading cultures in the compost, which again was pretty expected result here. So now we get into the interesting metrics that we can calculate with the new feature. Specifically average well color development, the diversity index, and evenness index. And so we're looking at SR1, which is the native soil, again in pink, and SR2 is the leaf compost in blue. And so the compost microbial community was much more rapidly able to adapt and utilize a wider range of substrates. And so looking at Shannon's diversity here, we can see that SR2, the leaf compost, had very high diversity much earlier in the assay, around about 10 hours, as opposed to the native soil, which took until around 15 hours to get to a similar diversity level. And this is reflected again in the evenness, where we can see a very high evenness index indicating uneven utilization of substrates around about that 12-hour time point, and then it falls off to 1, meaning that the curves are either reaching saturation, meaning that the substrates have all been utilized, or they've gotten up to the same level well before the native soil was even able to start metabolizing a few different substrates. So overall, this was fairly expected, again, based on the results from the previous slide, looking at the average replicates. But it's nice to have the confirmation here that the diversity and the evenness show that same lag. And again, the average well color development here we can use as a rough estimate of overall metabolic output of the communities in general across the 31 substrates on the EcoPlate. And we can see that SR2, the compost sample, was much more energetically active or metabolically active in this assay and was able to output much more energy overall than the native soil sample. So the next comparison that we did was looking at soil that had compost added to it, and then soil that had organic fertilizer added. That did not have any known biofertilizer cultures in it. And so the compost significantly increased the metabolic activity relative to the organic fertilizer. And so we can see that here on the average replicates, that for most of the substrates where there was metabolic activity, the compost addition soil MC1 here in pink showed a much higher signal, much earlier signal than the organic fertilizer in the soil. And just as an aside, this organic fertilizer in soil sample was taken from an apple tree that I bought from a nursery, and the compost in the soil was taken from a garden bed that had been neglected for a couple of years and had a lot of leaf litter decomposing in it. And so you can see we can take rather varied types of samples, at least when it comes to soil for these plates. So taking a little bit deeper look at this data, the average well color development was actually very similar for these two samples, even though the diversity and the number of substrates that were being used was different. We were able to see that for the most part, both samples did have approximately the same energy output by the end of the assay, while there was this initial pickup in the compost sample. And when we look at diversity, that's reflected again, where the diversity of substrates being used was much higher in the compost sample than it was in the organic fertilizer sample early on in the assay. And finally, the compost had consumed substrates about equally well compared to the organic fertilizer, as shown by the Shannon's evenness index. So while the compost may have been using more substrates or more varied substrates than the organic fertilizer, they were both using them at approximately the same rate and evenness, indicating a similar flexibility in metabolic profile for each of the communities. So just to summarize everything that I just went over, the simple workflow for the EcoPlates allows you to rapidly characterize many different samples and then analyze them very quickly. And there's not really a need here to worry about quantifying your cell titer whenever you have the average well color development to help you normalize and draw conclusions across your communities, as well as the Shannon's diversity and evenness index, which are powerful tools to allow you to make comparisons, even if you don't know about the starting cell titer. And you can also conveniently export all of those figures that I showed, as well as reports and raw data and any calculated parameters that you may generate for further analysis downstream in a platform of your choice. So adding Odin to your workflow so that you can run some of these assays. So first off, why Odin? Well, as Enrico and I have talked about, each well is going to exhibit a different rate of dye formation. So single endpoint reads that you might get with a traditional plate reader are not exactly ideal. And additionally, running a single plate at a time can lead to some issues when it comes to sample processing, availability of sample, et cetera. And so running multiple plates side by side, all collecting data simultaneously, is ideal when it comes to drawing these conclusions, because the more replicates you have and the more consistency you have in your sample preparation, the better, as you will be able to draw more confident conclusions and know that it's not sample prep or day-to-day variation that caused your significant results. And drawing these comparisons is very convenient and easy in Odin's software, which allows you to calculate a few different parameters along with all the community analysis features as shown here. The data output from Odin is extensive and can make the downstream analysis of your community data much easier. So now we have available to export as either CSV or JSON file types for your ease of use in any statistical software of your choosing. So overall, Odin makes your life easier when it comes to running EcoPlates and other plates, especially when you have multiple users or just one who has multiple samples to run. You would be surprised as to how fast the plates fill up in an instrument whenever you have an experiment running, because if you're running triplicates or multiple samples, that's automatically 6 plates. And if you have more than one user, they can disappear pretty fast. And so having all of your plates incubating at the same time, incubating in the same conditions, really lends itself to getting predictable and reproducible results. And then all of your data will be organized as projects within the software, which allows you to quickly and efficiently find and analyze your data. So I just want to go over a little bit about Biolog's ecosystem of products and services. We've talked a lot today about the phenotypic analysis for the EcoPlates, but I also wanted to mention that we do have a plethora of identification tools working off of this same microplate system, and powerful new phenotyping and identification software that is based on the same platform as the community analysis. We also have consumables that go with each of these different product lines with the identification. We have plate type for each of our different major classes of microbes, and we also offer customizable microplates. And finally, we have recently in the last year started our lab services, which now offers identification services in the form of DNA sequencing, MALDI-TOF, and phenotyping, as well as doing all the phenotypic analysis that you might need. And this lab is fully ISO certified and FDA registered for your peace of mind. So to wrap up, just wanted to emphasize that the microbial community analysis features here in the Odin software really give you the ability to listen to the voices of your microbial communities and condense them into something that we can compare one to another, even if we don't know exactly who is in that community or how many there are. And this is a really important concept in order to understand and leverage these soil and other environmental community samples and the data that we get from them. So with that, I'd like to say thank you and open it up to questions, which you can feel free to put in the chat, and we will get to as many as we can. And if not, we will be reaching out to you afterwards. Thank you.
Okay, great. Thanks, Max. So as Max mentioned, please submit your questions in the Q&A chat window. I see a number of them already have come in. And we also had a few questions, like I said, come in through the registration. So we will get started with those. There are a few of them are similar. So how do you recommend processing soil versus water samples to inoculate on the EcoPlate?
All right, I guess I'll take this one. So I think a soil sample is really important to being able to extract the microbial community efficiently from the soil matrix. And I saw that some people in the chat were asking if there is a standardized method, and so I can answer that as well. There's not really a standardized method because soils may be very different in their physiochemical characteristics. So you may be successful sometimes in using sodium chloride solution for that to extract the soil, and with some other more clayish soil, you may want to use a buffer. There is a buffer phosphate. So it really depends. And I always recommend to look in the literature for similar example to make sure that this is followed correctly. For the water sample, I think one good practice is to directly inoculate the water sample into the plates, and maybe after, depending on that, it also depends on the number of cells that you may have. If the number of cells is low, some people have used either centrifugation, which I don't recommend too much, or filtering, which is better to have more concentrated cells and inoculate them in body plates.
There was a specific question early on, Enrico, when you were talking about the EcoPlates. Do you put water in wells A1, A5, and A9, or your sample?
No. Yes, I saw that as well. No, you add your sample as it is. That's a really important well because it represents the baseline metabolic activity of your microbial community. It also tells you if there's some contamination from nutrients coming from your environmental matrix. So it's important to add the actual community there and hopefully have a signal that is not too high, if not absent.
Yeah, just to kind of go off of that a little bit, the other reason it's important to use your sample in those wells is because the Shannon's index and average well color development calculations utilize the negative control wells as a way to background subtract any noise from your data. So it's important to use your actual sample for those wells.
Right. There's another one about the sample, so I'll just ask those. Does salinity of soil impact EcoPlate's measurements?
It's a very good question, and yes, it does. It is important to, when you're working with a marine community, to dilute a little bit the sample. That's mainly because of calcium. Calcium can make, precipitation of the dye more likely. So there's also a paper in the literature discussing this very matter. So it is important to be mindful and maybe use dilution in that case and concentration of cells if the numbers are low.
We are getting some questions here about the Odin software demo and as well as some instrument cost questions and all those. We'll take those offline and follow up with you individually. But there are some more questions about the software specifically. So Max, let's see, I know there was one here. Hang on one second. When do you do the average well color development? Can you compare all substrates? Are there more sorry, I can't pronounce that. Substrates that generate higher dye reduction? Is there a need to normalize?
Yeah, so the average well color development is calculated across all 93 wells that contain substrate. So that's a generalized metric over the whole plate for energy output. So there's not really a need to normalize any further because it's already normalizing itself across the whole plate.
Great. Another question that we had was about the adjustments. So what are the adjustments you can make to the community analysis calculations?
Great question. So there are two main adjustments. There's a pattern development and a positive response cutoff. And so those allow you to tailor the Shannon's diversity and Shannon's evenness index calculations. So the pattern development is going to be the amount of total color development across the plate that has to occur before a pattern is considered to start. And so that allows you to clear up noise at the beginning of the Shannon's diversity index calculation. And the other one is the positive response cutoff. And so this allows you to customize the richness parameter in the Shannon's evenness calculation. And so richness, again, is the number of substrates that are being used in the plate that have a positive response. And so the positive response cutoff just allows you to set a threshold to which everything is considered positive. And so our default is, for instance, 0.25 OD, and that's the difference between OD 590 and 750. And so again, that one just allows you to clear up some noise in the first few hours of incubation.
Okay. There is a question about the data export. Can the data exported by Odin be read by OPM package or R language? I'm not sure that I understand that, but maybe you do, Max?
Yeah, yes. So the file is just going to be a CSV or a JSON file. So any computational software that can take those file types will be able to read them. The metadata and headers in the files are very simple. The architecture of the file is very straightforward, so you don't have to worry about any confusing inputs. Yeah, it's just a simple table.
Are there any examples using biolog carbon plates for proteases?
Proteins, yeah.
Sorry, go ahead.
I don't think EcoPlate has been used with directly with the protist. I don't think their redox dye would be ideal for this organism. I know that protist they have been studied using other biolog plates, not community, but specific protist to see their energy profile, their functional readout. So in theory, it's possible finding the right condition, but perhaps not using EcoPlates, but using other biolog solutions.
Yeah. Let me see another one. I'm interested in studying the effect of particular stress on soil microbiome of two regions and correlating with flora richness of the two regions over a period of 10 years. Using your platform, a single pot experiment can be set for it? Question mark.
Sorry, is that in the chat? I need to reread the question.
No, it was one of the ones that was submitted earlier.
Can you reread that, Kasia, please?
Yeah, absolutely. I am interested in studying the effect of a particular stress on a soil microbiome of two regions and correlating it with flora richness of two regions over a period of 10 years. Using your platform, a single pot experiment can be set for it? Question mark. I think it's might be translated a little off, but we can also always follow up offline because we have those in the registration if you want.
Yeah, so looking at stress response using the EcoPlates, you can definitely do that. So what you could do is add your stressor to the EcoPlate along with your sample. So if you want to add high salt or a drug or something like that, or some other compound that might be found in the soil, you can just add that to your sample on the EcoPlate and then use the community analysis features in order to look at some pairwise comparisons there as a pilot study, yes.
There was one. Can EcoPlates be used to identify and quantify named bacteria directly in a sugarcane juice?
Yeah, I think I saw this question. No, not directly, but yes, indirectly. There's evidence also in the scientific literature that people have inoculated a complex community into the EcoPlates and then incubated over time and just take the supernatant out of a specific wells where they have a signal and plated that to see if they could isolate something or directly sequence to see what could come out from that sequence. And the fact that the EcoPlate has 3 replicates also, you can pool 3 different wells in one sample and have a larger sample for either a plating approach or a DNA-based sequencing approach, if that's what you want to do. But directly, no. What the information you get from the EcoPlate is what that complex community functionality is, what it's capable of in terms of metabolic feature.
How can we use EcoPlates for marine organisms?
Well, I'll refer to the previous question that you can use it EcoPlate has been used with isolates as well. You have seen the example number 3, the study number 3 that I presented, that they were a single strain used. So you can do that if you want to characterize your strain. If you're working with marine organism, you may have to add a little bit of sodium chloride to reproduce the environmental condition that I used to. So you don't have to add too much, but a certain amount, 1 or 2% to the inoculation fluid mix where you have your strain, and that can be done.
Okay, perfect. Looks like we're running out of time here. So we did have a number of questions about the Odin software. As I mentioned, we will follow up offline. That's the new software, as Max stated, available on our Odin instruments, and we have different throughput systems available, as Max covered in a presentation. So we will reach out to everyone with those questions specifically and follow up directly. So thank you to everyone who joined us. I appreciate all the questions. I appreciate your time joining us. Thank you to our speakers, Enrico and Max. And if you have more questions, if you think of more, please submit them to the info@biolog.com. Otherwise, we will be sending their link through the recording to everyone who has registered, and you can always submit your questions later too. Thank you, everyone, and have a great day.